now in present mode. I hope that's okay. All right, great. So thank you for having me here today. And again, I apologize for the problems that we had yesterday. Today, I'm going to talk to you about what we do at Inari. I realize time is short and I don't want to spend too much time about explaining what we do at Inari, but I have a little bit of background as well. I guess within the context of this series of conference series, I think Inari is perhaps a good showcase also of the kind of data that is being generated, at least in the ag biotech, and especially the future trends of data that will be generated in the next couple of years by companies like us, but also almost certainly also by others. Right. So the title of my talk is a little bit lengthy. It's about so it's called Predictive 4.0. It's essentially about how we are integrating not only generating data, but also integrating machine learning models at Inari in order to be able to help us tackle some very specific biological challenges, which are essentially not only biological challenges, but also challenges in agriculture as a whole, right? So this is at the core of what we do. And before I move on, I'm going to talk to you about, are you still hearing me okay? Okay, so I'm going to talk to you about who we are and then what kind of technology we develop and then how we integrate into all that. So I'm not going to bother reading this. This is more of our core foundational mission. Essentially, we are what we call a seed design company. Means basically we're making better seeds for farmers. There's a lot of steps in between. There's a lot of technology that is fueling that. But the core of what we want to do basically is improving some of the crops that are currently being used across the globe for higher yield, less inputs, more sustainability, and so forth. So that's at the core of what we do. Unfortunately, I cannot go into the actual products today, talk about this, but I chose for today also to focus more on the data side, given the context of this conference. But if you have any questions, you can always ask. So very briefly, Inari was founded 5 years ago, relatively new, if you want, and we were founded in Cambridge, Massachusetts, as part of a flagship pioneering company. The good thing is, since last year, everyone knows who Flagship Pioneering is, because when I tell people that Moderna is also a flagship pioneering company, and people say, yes, Moderna from the vaccine, right? So this is a company that was founded with a couple of people, a lot of exploration in the first years. We have a very strong leadership team, people representing companies all across the industry, so the ag biotech industry. The slide is slightly outdated because we're actually more people now, and we just finished our C round funding, so we have a little bit more to go in terms of funding for the next couple of years. So we also have a very strong scientific advisory board that we're also strengthening with more computational people like Jonathan Pritchard, but some people might be familiar. I mean, these are very important people. Some of them have founded the company, also scientific co-founders. Jennifer Doudna has won the Nobel Prize last year also for her discoveries in CRISPR-Cas. And those people help us also in managing the scientific strategy that we want to go into. Okay, so what do we do? So essentially we came to the observation when we started that there are some major challenges also in terms of food supply, right? So growing population, increasing that you can grow crop. So in parallel to that, there was that are being developed over the last decade or more, and so these are either on the just basically on the editing side. So this is the CRISPR-Cas technology that has been talked about so much in the last couple of years, and for which Jennifer also won the Nobel Prize. This is essentially allowing us now to start modifying genomes efficient and high throughput manner, and also in multiplex way, so meaning different targets, right, and in different crops. At the same time, like I said, we need to reduce also inputs. So agriculture is pretty demanding in terms of resources that are needed. Water, fertilizer. Water can be scarce. Fertilizer is bad for environment. Therefore, we also look into some of the products that we can make that require less of those. And seed companies, of which there are not that many left, right? I'm just going to rapidly go through those slides because I want to get to the other bit on the data. So the impact that we want to make is increasing yield. I'm not sure what people's backgrounds are here, but if you are not in the agricultural sphere, the yield of per acre of different crops has been improving, and this is why we still have enough food, although our global population has tripled over the last 45, 50 years. It's not that more land mass is being used for agriculture, it's that yields have been increasing significantly. However, the rate at which they're increasing has been plateauing over the last couple of years. And that is basically because we've exhausted what we could do with the traditional technologies, and now we need additional technologies in order to, again, pick up the pace and follow global population growth. So the editing that I was talking about is one part of that. Computational part is another very important element that will help us to do that. So in the meantime, we also see that farmers, actually, it's really hard to be a farmer these days. They make less profit than they used to. We want to help farmers make more profit, so very important for us. And also, like I said before, we want more yield, and so we want more food supply, but we don't want to do it by using more land, right? Especially for soybean, this is something that's typically grown in areas where rainforest is also growing. So we want to avoid that. So we want to be able to do more with less, right? That's basically the bottom line. So like I said, there've been significantly, and important, like using best farming practices, so crop rotations is applied, and now there's a new gap that can be exploited in order to improve crops at a faster pace. So accelerating breeding timelines is one, increasing diversity of crop gene pools, and so forth. We are aware also of some societal questions around, what can be done, what cannot be done. It's very important for us to have those kind of involvements also from people from communities in society. But the bottom line is we want to improve crops, and the question is now, how do you do it? What are the technologies that we want to apply? So in order to explain that, I need to quickly explain to you what the difference is between how we did it and how we want to do it, right? So in the blue, see the traditional methods I was talking about. So I said before, in the mid-20th century, we started doing traditional breeding. Well, traditional breeding has been done since the last 10,000 years, since whenever people started to be sedentary. But in the mid-20th century, that became from like an art, it became a science. And statistics was used, trial and error approaches. It became much more rigorous than it was before, and that explained the significant yield boost at the time. And so, for example, soybean, there was an increase, but it wasn't massive, all things considered. So now we're flattening this off. So this is leveling off at 1% increase per year, which sounds like, it's not a lot, and it's probably not going to be sustainable if there's higher demand for food and for feed. So the other thing that was done until from the beginning of the 21st century until now is the so-called Gmos, right? So these are plants that have been made through what's now called the traditional transgenic techniques, where basically you can make plants that are resistant for herbicide or against the pest and so forth. So they've been super helpful in the past, but they've been mostly dealing with things like herbicide resistance and pest control. They're really expensive to make for whole regulatory reasons, but also scientific complexity that I'm not going to go into. And there was no really incentive also from companies to develop those to reduce inputs. So how is this different today? Two major elements, right? First one I'm going to talk about more. The second one I cannot talk about in this talk because I don't have time. So what we want to do is all right. So this means that we can use the tool to go into detail into a genome of a plant or even animals and model genes or sets of genes, elements that control genes, and so forth. This is something that we didn't have before. So compare it with your search and replace function from your Word, exactly what we can do now. Game changer technology basically allows you to rewrite the genome. Okay. The question that leads to that is then if you have this ability to do so, then what are you going to change in the genome, right? So for this, we need the predictive design. So this is where the computational part comes in. It's basically allowing us to, from all the genes in the genomes, and genomes can be really, really big. To give you an idea, in corn, so in maize, there's about 25,000 genes that are present in corn. We don't know about 1%. We know about 1% of what they do, the rest we don't. So how do you go from that to, with a tool that you can use to modify, to making a significant change that will give you more yield? So to do that, we need to develop those predictive designs. So it's not just about generating the knowledge and the data, but it's also being able to analyze them. So why do we need to be predictive? Well, because we cannot test one by one iteratively, right? We need to be as good as we can in predicting if you make these kind of changes, then your organism at the whole level will behave in this way. And so that's when we then say, okay, now we're going to make it. We don't want to make it and then see like trial and error like we did before on the left-hand panel. So that's why we need predictive design. Okay. So there's 4 components that I'm each going to talk about in this presentation, where we introduce, or we leverage some of those elements, right? So we use computational predictive design for computational breeding. So this is essentially how breeding was done before, very similar, except that you can predict with the tools that we develop, the algorithms that we develop, that you can predict performance of a hybrid before you actually make it. So what does that mean? Usually until 20 years ago, breeders would take plants that they like, they cross them, they look at the offspring, you get a whole variety of offspring, and then you take the offspring that you like and you do the same thing over and over again. So it's all trial and error, right? So you have to make the offspring and then you have to look at how they behave in the field. With performance, even made them. So that means basically you are genome from one plant, from another plant, and you're going to determine computationally how compatible they are to make good offspring. The advantage is obvious because it means that you don't have to actually make them. Computational analysis is always cheaper once you have it running than real world stuff. So it speeds up your breeding and you can develop, a base germplasm much faster than you would do if you go randomly in it. The second one is gene network levels. So this is about what I talked about when I said, well, there's plenty of genes out there, but you need to know which ones you're going to change because genes don't act alone. They work in different complex. And so you need to understand, how those complex function in order to unlock the value and then say, these are the ones we're going to change. These one we're going to leave alone. And then the other element on the right-hand panel is sequence diversity level. So this is where we basically correct, it's a step up from computational breeding, let's say. So you correct immediately some of the alleles, so some of the variants of the genes that are in the suboptimal form, and you make them optimal. I'll talk about it later, but it's essentially about being able to identify suboptimal form of a gene and then saying like, now this is the form that it should be, right? And then on the protein level, this is really, really amazing. This is using deep learning approaches to design optimal protein structures. I will come back to it. Okay, so data is only data, and models are usually much more useful than data. However, in order to have a model, you usually need also data to build it, right? So in my world, and lots of biotechnology, there are sort of four main layers, or five main layers. One of them is the genomes, which is your whole coding sequence, transcriptome, metabolome, proteome, and then phenotypes, right? They're all connected. Everything's encoded in the genome, but there are changes that can occur along the way, and not everything ends up in the proteome or in the phenotype. However, what we care about is the phenotype, right? In the end, and as much as I like genomes, at the end of the day, you want to see a phenotype that you like, and you want to be able to predict also how your phenotype is going to look like under certain circumstances. So this is just a primer, also adapted from a really nice paper from Virtual Lab on how does that work when you build a model. Essentially, you have your biological sequences. You build a testing model. Training essentially allows you, on a training data set, allows you to build this model that you can then run on real world, but data that you haven't used before, and then see how do they match up, right? This is the interpretation. So there's two ways of doing this. So you have your supervised learning and your unsupervised learning. Supervised learning is essentially when and you say, well, I want to find out what specific other marks are on the DNA sequences. You can obtain a model which essentially maps its predictors to target variables, right? So that's one way of doing it. Unsupervised learning is quite similar aim, right? But you are not telling the model which direction to go into, right? So that's why it's unsupervised. And so there's underlying that approach, there's different types of artificial neural networks, which are essentially based on inspired by biological neural networks in the way they work. But those are now used consistently across our society for many other things as well. But you can use those, and you can have machine learning problems also apply on biological problems. That's where it gets really interesting. So far, most of the advancements have been made in image recognition, which is also very useful. But the next step is sort of also having those set of filters or approaches to identify either what a sequence means in terms of function. If you can find optimal sequences without having an optimal sequence in mind, those kind of things. So there's some challenges which I think are relevant for this talk as well. So the data challenge is major in plants. So plants are not humans. Humans have, in terms of research, there's a lot more research going into one species, the human species, than going into any of the plant species, or I would even argue combined, right? So quality of your data, quantity, diversity is a problem. And you need good data to make good training models. And then also some of the tools that we struggle with. Some tools are developed for human use, so human data use, but are not adapted for plants. And then we always have to retool them in order to get them to work, which takes time and resources. Okay. So the second is the first thing I want to really talk about is at the plant level. So the computational breeding, I mentioned it a little bit before. So genomic selection is something that is used, and variotyping is something that's been used for a couple of years now. On genetic data from parents, and you can then say, well, parents would make good offspring, these ones not so good, before you make it, right? And that's very important. So it's actually quite accurate now. So in our hands also, we have up to 94% accuracy. So what does that mean? That means basically when we make 10 predictions, 7 out of 10 at least are going to be indeed superior, which means that this model kind of works. Not kind of, really works, right? So you predict best parental combinations for best performance, and then also it needs to be tied to the right environment. But this is really important because it helps you to develop much faster lines than you could if you were to breed them. Right? Okay. Then on the next slide, so this is the second component I talked about. Many genes out there in plants are known. It's going to take a very long time before, if ever, we figure out what all of them do at any given time in plant development. So how do we go from that to then say, well, we're going to pick this guy, and this guy to make some changes in the plant? So what we do is we also build some models around gene function, and more specifically about specific regulatory circuits. So plants are very much like any kind of engineering problem where you want to break it down in smaller pieces of the problem and then figure out, what do I need to change and what is the consequence of the change, right, for the improvement. So that's kind of what we do at a smaller level than plants. So we go into single cells. So this is also something that's a new technology that's opening up a lot of doors that were closed before. So single cell technology allows you to go into a single cell, map out specific regulatory networks, make some changes to the networks, see what happens, and then to decide, this is what I'm going to change when I go into the plants. And so the time at which this happens the turnaround can be real quick. And that's very important for plants, right? Because plants take a long time to grow. So for example, we've done this for a trait that we call nitrogen use efficiency. So this is a trait that we work on so that plants require less fertilizer and give you the same yield. So very important from the sustainability point of view. There are circuits that regulate how plants process nitrogen. And so if by understanding those circuits and by modifying them in the right way, you can improve how the plant uses the nitrogen. That's what we do here. On the right-hand panel, you see how we then apply that model and the output of the model. And then we develop a whole set of plants. So there's no number on there in terms of numbers yet, but it's still pretty much hit or miss, right? But at least it's better than doing it random. So the data that we use here are also based, like what I said before. We have training data and samples that we use, and then we use that also to predict and test for. So why is this important? Because at the end of the day, we don't care so much about how the genes are expressed, but we really care about what's on the right-hand side. And that's basically, the yield. We see those numbers, that's the yield of your plant. And so if we can go from a DNA sequence or any data set that we can quickly access and measure to an accurate prediction of the end result, i.e., being yield or plant performance, that is extremely important for us because everything in between and a lot of resources. So number 3 here is sort of the upgrade that I mentioned before. So without going into too much detail here, essentially there's a lot of genes that are in plants that are in any organism that are in the suboptimal form, right? Call them alleles. So you can have specific forms that are better. You can also have what we call deleterious mutations. So plants that carry deleterious mutations are not necessarily sick. They're just not as optimal as they would be if they wouldn't have that very specific form of the gene. So, but of course, not all genes are suboptimal. So how do you find the same problem as before? How do you find the genes that have these suboptimal forms? How do you predict what the optimal form is and then change it, right? The first part of that question is computational question, for which we have developed also computational filters that will allow us to identify sets of genes of which we think they are not in the right allele, not the optimal allele. And if we would have technology to actually go and edit those, the plants would be much more performing. Of course, not by just one modification, not just one gene, but the additive effect of many, many genes, right? So hundreds of genes. Again, something that if you know what you need to do, you can accelerate your timelines to improve much faster than what you could do before the 21st century. Right? So more of this is a little bit more detail. So we call it breeding by editing, right? So that we have a long-term goal. Like I said, we have 80 million possible targets in corn genome alone. So if so are either built or adapted for 10,000, hopefully thousands, it's a much more manageable number to start modifying and to see outcome. And so if we use that model, going back to the first example that I showed on predictive breeding models, so we apply those filters and we say, well, if you would select plants that have the optimal alleles, how much impact would that give you on an increase by 15%, which is really, really important because it helps also predicting which parents you want to combine. Okay, so this is the overview. There's also a really challenging part in terms of once you have those thousands edits, how do you actually edit them? Because I said before in the beginning, I said, if you remember, I said, well, we have the tool now to edit and multiple edits. That's all true, of course, but there's a difference between a handful of edits and 1,000 edits. Right? Working on that. So the last part I want to talk about is actually what I find one of the most exciting developments in the last couple of years. So this is a project that we call PEDAL. It's protein design using deep learning. So you might have heard from Google 2020. They had Alphabet DeepMind published work also on predicting protein folding. So this is AlphaFold. So this is a tool that can tell you how a protein is folding without ever have seen the protein. So until we had that tool, that algorithm, people had to do crystallography to then see how does the protein look like. Now this algorithm can do that without even ever looking at it. And it's really, really accurate. So that's one mind-blowing thing. But we actually take it a little bit of a step further because we want to be able to develop also optimal forms of proteins, not only of genes, right? I talked about before. So for me to explain this, I just need to explain the concept here. So what you're looking at are essentially on the left-hand panel, you can higher through evolution because evolution selects for improved fitness, not for decreased fitness. That's why you can never go back to those. Basically means if you would screen every single organism in the world, all of the proteins would look like the ones that are in that on that mountain in the green dots. But it doesn't mean that there's other more optimal forms of that protein. They just don't exist because they haven't evolved that way, right? So this is what on the right hand panel, the predicted protein there in the orange dot represents. So how do you go from that to that? Well, you can do that again by having a computational model, and that has been developed also and also been shown. So there's been a paper from the George Church Lab, who's also in our advisory board, and they have developed a model, deep learning model that sort of what could have happened from an evolutionary point of view, and then spits out a number of protein variants that don't exist in nature, but that it think is the best version. And so those have been tested experimentally on this green fluorescent protein. It's a protein that comes from marine organisms, and gives you fluorescence. and they put that through the algorithm, and they came out with a design that's actually much better than what is out there in the real world. All by computational analysis and prediction, none by actual experimental searching and then feeding it back into the model, right? So this is really the game changer technology. So that means that for lots of the genes that we have, the proteins that we have today, we can use that potentially to then start developing new and improved proteins, or at least predictions of new proteins that will improve fitness of the plants. And now we also have the tool to do the editing, so the editing tool to then go into the plant and make the necessary modifications. Basically accelerating evolution or redirecting evolution into a form where it hasn't gone into, right? Okay, so this is what I explained also on the correlation. I'm just going to hurry up a little bit. So yeah, so major game changer also, data will also be important here. The learning data sets are what defines how good your model works. And developing such a model will greatly increase the speed at which we can develop superior or improved plants, right? Okay, so with that, I just want to stop because I know I'm a little bit over time. I want to thank the people here for inviting me. Activities, and yeah, I'm happy to take.
Okay, thanks, Fred, for this excellent presentation and overview of the activities at Inari. I think it was quite interesting. So now we have some time for questions. I don't know if anyone wants to start. So
maybe I can start with the question. So you talk about the use of neural networks. One thing that has been a recent trend is the fact that neural networks are opaque, so there's the need to provide a sort of explainability to artificial intelligence approaches. Is that a problem you faced in this sector?
It can be. It depends how much attention you pay to it. So let's say we want to, our prime objective is to have something as an output that we can use. Understanding exactly how it works is not the prime objective, but of course we want to be able to replicate it and also to sort of evaluate how good the output is before we start testing it. And that requires you to know a little bit what's under the hood. And so black box approaches are difficult in that regard because you don't really know how well the output can be, right? Because there can be biases, right? That has been shown with a lot of deep learning approaches also and the black box sort of model, depending on the input that is provided for the training model. And so if you understand a little bit better also how the algorithm works, you can also investigate some of those biases and improve the quality of the output.
So, and is this something you have taken concrete steps to address?
No, right, because we wanna make sure that we can put the things right away. Pretty cautious in general and wait until you fully understand what you're gonna do in an organism.
Thank you.
okay, do we have any more questions from the audience? I have a difficult question to you. That I know that although it is very difficult to quantify, but since the major focus of this workshop is on data, I will dare to ask you, even I know beforehand that the answer is difficult, how much of this reduction in the seed development cycle, let's say, would come just from data integration models without genome editing or
So they both go hand in hand, right? So the one needs to come with the other, and especially I think the editing, we wouldn't be able to do much with the editing if we wouldn't be able to apply any of those models longer term. The opposites might be less true because, like for what I mentioned on genomic prediction for breeding, you don't really need to do editing on that. It just helps to be able to identify which parents you want to pair together. So I'll just do the first part. So if regardless of the editing, if we wouldn't have data to support the genomic prediction models, which don't require editing, we would have had a major problem also in improving crops over the last 20 years. So it is now consistently used, and I would argue that there are very few breeders out there or breeding companies that don't use in some way or another genomic prediction pipelines, and therefore all the underlying data that is being used. Yeah, that is for sure the case. I think for the editing, like I said, we will need it in order to be able to do significant editing, and I mean editing to have significant changes. How much? Well, I think it's almost to say, like, if we wouldn't have any data or models derived from the data that we either obtain or generate, I think we would be able to do 10% of the things that we want to do with editing. Yeah.
Okay, thank you. So, any more questions? If not, I think I will give the word to Hannes for the next section, session, and I would like to thank again Fred for your participation in this workshop and your contribution.
Yeah, thank you very much for having me.
Actually, I had a question. It might be a stupid one. So hopefully it is not a stupid one, but we'll see. So one question, because you just said that you obtain or generate the data. Can you elaborate on that a little bit? Because I can hardly believe that it's really easy to generate this kind of data as even like as a kind of young company like you are. So where does the data come from with the quality that you need in order to train these kind of deep neural networks?
So it's a really good question because I didn't explicitly mention it, but I think another element that converged with the development of new technologies was also the significant drop in cost of genomic data, right? So this is something that started when the first genome was sequenced, and it dropped massively over the last 20 years as well, which means it's far cheaper to generate data now than it was before. In fact, in some cases, you're almost at the point where it's cheaper to keep a sample and generate an additional set of data than to store it somewhere on a drive. We're almost at that point. So I exaggerate a little bit, but it's not that far. And so we don't generate all of our data ourselves because, like you said, there's a lot of things out there that would take a lot of time. We do in some cases, but there's also a lot of public data sets out there that we can use. And then we have also partners that in some cases have their own data sets on genetics that they've used that we can also integrate. But there is certainly an element of data generation that is becoming increasingly important for us. And in fact, we actually have a position open for a VP for computational sciences, just because it's becoming more and more important. And so we do foresee also in the next couple of years that we will generate much more data that we have. But again, I think it's not so much about, you know, how much data do you generate, because it's easy to generate a lot of data. It's more about what is the relevant data set that you want to have to answer your question, right? There's that. And then there is also what we have observed across public data sets, right? And it's not to criticize public data sets, I mean, or public institutes that create those. It's just that there is no clear and on, you know, descriptions of what they've generated, how it was generated, and so forth. And which means that if we actually have done this, if we take data from an experiment that was done across in the same plants for same conditions from many different labs that are deposited in databases, and we compare those, we see huge amounts of variation across those data sets, which you wouldn't expect. And that's just because either, you know, the metadata is not right, or the data wasn't curated properly, or the QC was done differently, you know, all of those things. So it makes it really challenging to say, well, I'm just going to mine the entire public data set. You might find a lot of things, but I mean, your output is just as good